Hello everybody, I am Dr. Sandeep Pandey, working as faculty Center for Botany of the Pratap Singh University, Rewa, Madhya Pradesh, India. Today, I am going to discuss the topic potentiality of medicinal plants against SARS-CoV-2 virus. Global epidemic COVID-19 has emerged as a big challenge for medics to find remedies for further spread and control. More than 30 million cases with up to nearly 5 lakhs casualties has been witnessed globally due to corona epidemics. The chemical drugs have been used widely during COVID-19 infection. However, their prolonged use causes impairment in immune compromised patients. Molecular docking techniques based on analyzing binding affinity between ligands and virus protein have set up a good platform in discovering new drugs. The natural compounds has emerged as better alternative compared to reference drugs in minimizing infections. Recently, World Health Organization has permitted research on phytochemicals and their potentiality in form of natural drugs to control COVID-19 infections. Now, these are the methodologies of the research work. Plant-derived compounds has been studied for their efficacy against SARS-CoV-2 virus. SARS-CoV-2 main protein ID6LU7 has been selected from protein database and used as target protein. PubChem database has been employed for identifying and searching plant-derived compounds with affinity to target SARS-CoV-2 main protein. Remdesivir, whose identification number is CID 1213040116, was used as a reference drug to compare the interaction between plant-derived ligands and various main protein. The studied compound were analyzed using SysAdmi tool on the basis of following parameters. Number one, drug likeness, physicochemical properties, water solubility, pharmacokinetics, medicinal chemistry and Swiss target predictions probability, and molecular properties. Pyrex, virtual screening molecular docking tool designed with VINA, has been used to analyze binding affinity and interaction between the target protein and plant-derived ligands. Altogether, 10 chemical compounds have been identified to target SARS-CoV-2 main protein. As shown in the table, these are the ID number of the compounds. These are the scientific name of the plant from which these compounds has been derived, and these are the common or local Indian name of the compound. Now the results. Number one, drug likeness of compound. All the compounds follows Lipinski and Egan rule, and they have the similar bioavailability score, that is 0.55. Next is the physicochemical properties of the compound. As shown in the table, the physicochemical properties should be optimum as it is an important parameter during drug discovery process. Now, these are the physicochemical properties of the compound. The important parameters are the number of heavy atoms, then number of aromatic heavy atoms, then fraction CSP3, the number of rotable bonds, the number of hydrogen bond acceptor. This is the important parameter for any compound. Then number of hydrogen bond donors, it is also an important parameter. Then the molecular reflectivity and topological polar surface area. Now this table, it shows the lipophilicity of the compound. The lipophilicity of the molecule affects the solubility and permeability through the membrane. These are the various parameters on the basis of which the lipophilicity properties of the compound has been analyzed. These are the various values for all the 10 compounds. These values have been generated using SysAdmi website. Next is the water solubility of the compounds. The water solubility property of the compound is based on three parameters: log S, log SALI, and log SSLI COSIT. These are the solubility values of all the 10 compounds. These are the classes for each of the parameters, subparameters. These are the solubility values of all the compound. Again, these are the classes of this subparameter, solubility classes. This is the subparameter for the water solubility for all the 10 compounds. These are the various values that has been generated using Swiss ADME website. Now, these are the classes of solubility. Now, next is the pharmacokinetics of the compounds. The pharmacokinetic data of the compound determines absorption potential of any drug. The gastrointestinal absorption property of all the compounds is very high. The blood-brain barrier permeability, as shown in the figure, 6 compounds shows blood-brain barrier permeability, and 4 compounds do not show blood-brain barrier permeability. 3 compounds acts as PGP substrate. Then, except 2 compounds, 8 compounds does not inhibit CYP1A2 cytochrome. Except 1 compound, 9 compound does not inhibit CYP2C19 cytochrome. Except 2 compound, 8 compounds does not inhibit CYP2C9 cytochrome. Except 1 compound, 9 compound do not inhibit CYP2D6 cytochrome. Except 1 compound, 9 compound does not inhibit CYP3A4 cytochrome. Similarly, skin permeation value was found to be high for cardamom and least for the compound alene. Next is the medicinal chemistry and Swiss target predictions probability of the compound. Medicinal compound predict that plumbagin shows 2 pain alerts and other compound do not show pain alerts. 4 compound do not violate Blank law, whereas 6 compound violates Blank law with 1 alert each. Now, 6 compound shows no lead likeness with 1 violation. 3 compound shows no lead likeness with 2 violation. 1 compound, phyllanthrin, shows no lead likeness with 3 violation. Gymnostrogenin shows maximum synthetic accessibility and eugenol shows minimum synthetic accessibility rate. The target probability score is less. It means that compound have high target attraction. Now this table, it shows the molecular properties of the compound. These are the molecular weights which has been generated using Swiss ADME database. Then these are the log P values. These are the rotable bonds. These are the acceptors. These are the donors. And this is the surface area of all the compounds. Next is this table. It shows the interaction between SARS-CoV-2 main protein and plant-derived ligands. All the 10 compounds has been compared with the standard drug remdesivir. The molecular docking between SARS-CoV-2 main protein and ligand generated negative value for free energy in the grid box. The free molecular docking score reported to be remarkable in case of methanolide A and followed by plumbagin and A-cyperon when compared with the standard drug remdesivir. Now, as shown in this figure, this is the 6LU7 protein. This is the target protein, the SARS-CoV-2 main protein, and these are the 3 important natural products with the remarkable molecular docking score. These are the docking results. Next is the conclusion. The strength of attractive force between protein and ligand determines molecular docking score. The energy score makes a compound more effective against target protein. In this study, all the phytochemicals shows remarkable molecular scoring value 4.3 to 6.3 kilocalorie per mole when compared to standard drug remdesivir. Based on these predictions, 3 compounds, methanolide A, with the molecular score of minus 6.3 kilocalorie per mole, followed by plumbagin and acipron shows better potentiality in targeting SARS-CoV-2 main protein. Thus, there is a need of further experimental validation of these natural compounds for their wider applications in drug preparations against COVID-19 virus. These are the certain references which has been cited to the literature. Thank you. Now, if there are any queries, any questions, you are please most welcome to raise any queries or the questions.